Hello, welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. So, this is lecture 22 where I will discuss the metabolic engineering case studies of early steps of indole alkaloid biosynthesis. So, let us go to the concept to be covered. So, where we will see overexpression of TDC and STR genes in tobacco, overexpression of TDC and STR genes in periwinkle, and then expression of periwinkle genes in heterologous system, and then hairy root culture as a system for indole alkaloid production, and if time permits we will talk about overexpression of transcription factor genes in periwinkle. So, let us go to the board. So, first one is this, that let me very briefly work out the pathway what we have just studied. So, important point is this, that tryptophan, we start with tryptophan. So, I will write indole alkaloid biosynthesis and pathway manipulation. So, what we said that tryptophan makes tryptamine, and then this joins with secologanin, and makes strictocidin, and strictocidin in the next step it makes strictocidin glycon, and from there different products form such as cathinamine, stemadenin, ajmalicin, and so on. So, this route comes from terpenoid, and this is from the secimate chorismates, that means so the enzyme which is responsible for this is tryptophan decarboxylase, which I have mentioned, and the joining enzyme is STRs, that means strictocidin synthase, and this one is this SGD, strictocidin glucosidase. So, now we will see this. So, first of all the scientist attempted to make cell cultures of Catharanthus roseus, the English name is periwinkle, and what they have done that they transform the cell cultures with TDC and STR. So, that means expression of TDC in cell culture of Catharanthus roseus and expression of STR in cell culture of cirrhosis. So, when TDC was expressed, so it produces tryptamine in higher amount, but that did not affect the or that did not uplift the synthesis of strictocidin, because the secologanin may be a limiting factor there. So, that means expression of TDC only upliftment of tryptamine, whereas when STR was overexpressed, that leads to formation of upliftment of strictocidin in marginal amount. So, what happens in this case, an enhanced STR activity was detected, but strictocidin level was only marginally increased. This is what happened with the cell culture. From there, the scientist, so they came to a conclusion that that only the overexpression of the genes of the same pathway is not enough. Perhaps the regulation lies in further upstream of the pathway, or there may be the transcription factors which need to be modulated, so that the product can be produced in higher amounts. So, these are the several questions which were subsequently raised. So, when they have used, now they have used the tobacco system. So, when they have used the tobacco, actually it is not the proper tobacco, I should write the Nicotiana rustica, a species of target So, here both TDC and STR genes were transferred to Nicotiana rustica through agrobacterium mediated transformation, and transgenic plants were raised, and what has been found that transgenic plants are basically producing both TDC as well as STR, and these activities were successfully detected. When that is so, then it is important to see that what happened to the product. So, tryptamine, yes, tryptamine detected, but no strictocidin, sorry, no So, then what they have done, feeding of secologanin can now help in production of strictocidin production by the transgenic plants. So, why? Because tobacco is not meant for, or Nicotiana rustica is not meant for producing the indole alkaloids, but genes can successfully be transferred, and good point is that TDC substrate is tryptophan. Tryptophan is universally produced aromatic amino acid. So, when TDC was expressed, so it can take the endogenous tryptophan available in the pathway, and that leads to the upliftment of tryptamine. Okay. However, the tobacco, transgenic tobacco cannot make strictocidin, because although a terpenoid pathway is operating there, but it is not producing any secologanin. Therefore, in order to produce strictocidin in Nicotiana rustica, one has to feed the secologanin, and exactly they did, and as a result of that the transgenic plants produced strictocidin. So, this gives confidence to the scientists that indeed the this heterologous system can be utilized for producing some of the early pathway alkaloid products, even in in the heterologous system like tobacco. So, that is the good thing. So, this actually gives some confidence to the scientists. So, then they move further to explore the system. So, now the next case study what I am going to cover, I need to draw this pathway again, but anyway that is required, that is the biotransformation of tryptamine and secologanin into plant terpenoid indole alkaloids by transgenic yeast. So, this is one of the very interesting case studies which I am going to discuss now, and this paper was published by Gardlings et al. So, in the year of 2001 from Netherlands, Leiden. The group leader was Robert Verpoorte, PWRT, and this was published in Applied Microbiology, Biotechnology, and the volume number 56, page number 420 to 424. So, let us see what they have done. So, we have to draw the same pathway again, that means the tryptophan makes tryptamine, and then here comes secologanin, and these two joins and make strictocidin, and strictocidin aglycone, sorry. And then different products can be produced, such as cathenamine, or azmalicin. Now, if I join with arrow, uh, so the source genes are STR and SGD from Catharanthus roseus, and the target organism, it is a eukaryotic system, that is this Saccharomyces cerevisiae. I just write yeast. So, what they have done is basically they made a construct containing both STR and SGD, and then so basically the cDNA which encodes for strictocidin synthase and strictocidin glucosidase. So, those genes were used and the constructs were prepared under proper control of promoter and terminator, and then yeast cells were transformed with these cells. So, that means what if we write here that yeast cells transformed with STR and SGD. Okay. So, let us consider that transgenic yeasts are produced and these yeasts express STR and SGD. Now, what is going to happen? So, yeast requires growth in a medium. So, these genes are there, these genes can be detected. Okay, even the expression can be studied, that means the mRNA produced can be measured, but product formation most likely will not be there, because tryptamine and cycloganin are not available. So, what is required? Basically that you have to do feeding. Feeding of substrates. What are these substrates? One is the tryptamine and cycloganin. Now, when this feeding was done, then what has happened, that yeast successfully accepted tryptamine and cycloganin and produces strictocidin. Now, this strictocidin, what is produced in the yeast cell, this strictocidin leached out into the medium. So, strictocidin maximum amount detected in the medium. So, that means it is leaching out or permearization of strictocidin outside the yeast cell, because too much strictocidin is not good for the yeast, because yeast are not meant for producing strictocidin, but it is in the medium. But what happens, that with time the strictocidin in the medium getting disappeared, and as a result of that what happened, that scientists they have detected of cathinamine. Okay. So, and also a little bit of azmalacin. Now, why it is happening? So, although the genes for STR and SGD are expressed inside the cell, so it appears that the strictocytine glucosidase that is basically that is again secreting out from the yeast cell, and it is basically localized in the medium. So, what happens that when strictocytine are produced inside the cell, so if we make a light drawing, this is the yeast cell. So, as a result of STR, strictocytine is produced, and that is coming out in the medium, and where this is converted into catenamin. That means, the genes for SGD is basically coming outside the medium. So, that means that SGD is basically there. So, what happens that when you do the feeding with tryptamine and secologanin, ultimately you are finding a good amount of strictocytine in the medium, and then with time that is getting converted into catenamin, and that is because of the SGD activity. From there, it is clear that expressing plant pathway genes in yeast is feasible, and it is possible to produce the product by yeast cells, and these products are leached out into the medium. So, which makes the downstream process easier, so that these alkaloids can be easily extracted out. Otherwise, if it is located inside the cell, then you have to break the cells, that is energy dependent process, and get the things out. So, this is a very good system. Now, this is fine. Now, one important point, which is this, the cost of tryptamine and secologanin. So, tryptamine is not that expensive, but secologanin is very expensive. So, if you want to make a process at the commercial level, so this may not be feasible, but tryptamine is not that much expensive. Therefore, scientists they look for alternative sources of secologanin, and they finally found a plant which is called snowberry. The snowberry fruit, it is white in color. Maybe one of the subsequent classes I will show you the photograph of snowberry. Strawberry fruit is basically appears to be a good source for secologanin. The scientific name of strawberry is Symphoricarpus, SYMPHORICARPUS, Symphoricarpus albus, ALBUS. So, strawberry fruits basically these are the berry botanically. So, the fruit extract contains a huge amount of secologanin. So, this has basically two advantage. What are these? Number one, fruit is basically a source of sugar. So, that means the sugar what normally one added in the cell as a carbon source for growth, that can be reduced. And number two, the fruit extract contains also secologanin. Therefore, growing the transgenic yeast in a medium which is enriched with snowberry fruit extract, which caters the need for sugar and cycloguanine, only you have to do feeding of tryptamine, and that basically leads to the formation of your alkaloids, that means cathinamine, and this cathinamine can be converted into azmalisine. So, this is a beautiful case study where early Catharanthus pathway genes are transferred into the yeast, and as a result of that yeast, transgenic yeast can produce the early indole alkaloids, which is produced in the plant. Now, let me go to the next slide. So, that means we will see the hairy root system. We will discuss little bit about the hairy root system for hairy root system of Catharanthus roseus for indole alkaloid production. Now, what I have not said, I have not discussed fully the late steps of indole alkaloid biosynthesis. So, before I discuss this, it is important to know that strictocidin glucoside, ultimately what it produces? It produces different products like catharanthine and vindoline. So, scientists, and then what I have said, that these two joins and produced vinblastine. So, scientists they attempted to use hairy root system for studying the indole alkaloid biosynthesis and regulation. Why hairy root? Because hairy root is a sturdy system and it is genetically stable, therefore, manipulations can be done. So, here there are a few aspects which need to be discussed before I come to the hairy root. So, it is important that we should also know the what happens to the normal shoot cultures, whether shoot cultures of C. roseus can produce indole alkaloids, whether cell culture of C. roseus can produce indole alkaloids, whether there is any role of light in alkaloid synthesis, and any role of green tissue in alkaloid synthesis. So, this needs to be addressed. So, once we address this, then it will be better to bring the hairy root system in the in this context. So, in the next class we will talk about that. So, I will end this class here. Thank you very much.